And our next talk will be given by Neo from Mike Malin's lab in London, and he's using the iPSC system to screen for novel cofactors and inhibitors of HIV. And yeah, good luck.
First, but bear with me. So my grandfather attained his PhD in the 60s. He died when he was 35, and when my dad was 10. I was subsequently named after him, and learning about his life really inspired me to pursue this route. My grandmother gave me a bag of coins that he had collected. One of them was an American silver dollar from 1921. And I promised her that I'd have it in my pocket today when I gave this talk. All of this is to show my gratitude for the chance and the opportunity to talk here in front of you today. So without further ado, this is my presentation on my PhD project using iPSC natural variation to define HIV regulatory networks. So this project aims to leverage the genetic diversity of iPSCs to uncover novel regulatory factors influencing HIV replication. By doing this, we can study the complex network of cellular factors that the virus interacts with. Previous studies have looked at knockdowns, knockouts, cDNA overexpression, and protein interaction studies, and so on. The approach that we're taking here will enable us to capture smaller effect sizes. It will enable us to look at transient interactions reflective of natural infection levels, and also give the advantage of identifying factors related to cellular health, such as viability and proliferation. So iPSCs are a variety of stem cells reprogrammed and generated from terminally differentiated somatic cell lineages. They're able to self-renew, meaning that they are a virtually inexhaustible resource, and they have the ability of differentiation to almost all cell types with the exception of placenta tissue. So the main differences between these lines can be attributed to the genetic background from the donors from which they were derived. This is due to the nature of their generation, which basically confers a transcriptionally similar state within all of these lines. A paper from Charles Rice's lab also found that these lines intrinsically express many interferon-stimulated genes that varies in a cell type specific manner. So as these cells differentiate, they lose this expression of ISGs. So we've got a program at King's which has 3 arms, which all tackle distinct but related questions. My own work, which is the middle lane here, involves screening 150 iPSC lines with challenging them with vectors and virus to obtain infection phenotypes for each one of them. Each of these lines has genomic data attached to it, as well as transcriptomic data taken from the time of generation, as well as the time of infection. The aim is to integrate the genetic profiles of these lines with their infection phenotypes to uncover factors that could underpin the differences that we see between the phenotypes of the most permissive and least permissive lines. On the right here, you can see another project which took a subset of the lines that I worked with in this biobank of iPSCs, differentiated them into macrophages, characterized them, and challenged them in a very similar manner. On the right is a more clinical-based project, which took HIV-positive blood samples from a pre-ART era and reprogrammed them into iPSCs through the CD4 negative population. So these are HIV-free. These all have clinically defined phenotypes ranging from elite controllers to rapid progressors, viremic controllers. And these were challenged again in a likewise manner to what I've described. Both of these projects also had their own associated omic data sets to integrate into their phenotype, infection phenotypes. So there were 150 lines, and we approached this by using 5 different assays. 2 were vector-based, looking at HIV1 and 2 vectors. 2 were TZM-based, looking at NL43 and ROD-TZM for HIV1 and HIV2 in these assays. And we had a full-length HIV assay as well, looking at NL43. So essentially, the vectors and virus would give us phenotypes based upon the events of up until integration, at which point a microscopy-based assay would take over, giving us phenotypes based upon the translation of viral DNA into viral protein by looking at stained GAG. And then the TZM assays would give us phenotypes based upon the progeny infectivity of these lines. So Here's an example plot of one of the run-throughs of my screen. So you can see here our 293T positive control, and then beneath it, you can see 10 iPSC lines of varying permissivity. So at the start of our data analysis pipeline, what we did is we approached this with a linear regression model where we would take the linear-most portion of this curve and use that to calculate an average titer for each line. However, we noticed that depending on which portion you decided was the linear point of the curve, this could alter the titer somewhat. So we applied additional statistical models that could be fitted algorithmically. And these included the logistic model, a logarithmic model, and an area under the curve model. And these models include all of the data points in the curve in their calculations. But it's likely that no one model is going to be sufficient in explaining the complex nature of the interaction between host and pathogen. So therefore, it's likely more applicable to use all these models in conjunction with one another to give us more confidence in characterizing our extremes of the phenotypic spectrum. So this is an example of the vector data of the HIV-1 vector data from 150 different lines. And you can see our positive control 293Ts up there. These lines were screened about 10 lines per week, and it's shown here by the linear regression model in this case. So this data seen here has been normalized by Z-score, transforming it into a normal distribution, which allows us to compare data from different weeks on the same scale. So each point in this graph is a single cell line made up of 4 replicates, at least 4 replicates, some had more. And we can see from this data that there are clear differences from one side of the spectrum to the other in terms of permissivity, and we see 10-fold differences from one end to the other. So this demonstrates a clear spectrum of susceptibility in this data set. We have similar plots like this for every other one of the statistical models that we also utilized in this study. So you've got 4 models, you've got at least 4 replicates, so you've essentially end up with 16 data points for each of the cell lines that you're using here. And when you want to draw that many dimensions on a 2D surface, it's kind of impossible. So we turn to dimensionality reduction methods such as UMAP, which calculates similarity scores for each one of these lines, then clusters based on those scores. So we also used k-means clustering as a mathematical approach to distinguishing between the phenotypes of our lines, rather than just choosing an arbitrary number of the top 10, the bottom 10. So this UMAP that you see here it shows all of the different assays and models combined into one plot. And you can see from this plot in the blue is our permissive lines, and in the purple is our our least permissive lines. And if you look at these ANOVA down here, you can see that regardless of where they are in these clusters, they're all statistically significant for one another. And the data points within the actual ANOVAs are quite tightly grouped together, suggesting that all the models are in agreement with one another that these extremes are in fact extreme in this data set. So we have 5 assays. So we end up with essentially what is 5 different rank order lists, one for each assay. But we're interested in how do the different assays correlate with each other across the extremes. And here is what is essentially an overly complicated Venn diagram. But what it shows you is the permissive lines in this case, the most permissive extremes of each assay, and how the different data sets correlate amongst each other in terms of the rank order. So you can see that the vector data clusters quite close together with many cell lines in common between them. The same is true for the TZM assays here. And we also have cell lines going across between the different assays from vectors to virus, virus to TZM, TZM to vectors. So what we're seeing here essentially is correlation between early and late events, but also between HIV types. And this is the same data, but for the least permissive lines. So this is a correlation matrix showing the transcriptomes of each one of these lines. And you can see that we have a group of least permissive and a group of most permissive lines, and how similar their transcriptomes are to one another. There's a difference of about 5% on average between the transcriptomes of these completely opposite phenotypic lines. So they are quite similar to one another. And this 5% difference between the transcriptomes is where our differentially expressed genes lie. So you can see in this heat map here is a pairwise comparison between a permissive line and a refractory line. And you can see that there are differentially expressed genes between them. You can also see in this volcano plot here associated with this heat map that we do find some well-known ISGs popping up as differentially expressed between them, albeit at a low fold change. So out of the 19,500 genes within this this comparison, about 2,000 of them were characterized as differentially expressed, but only 301 were showed a log fold 2 change of greater than 2, and at the same time being statistically significant at an alpha of 0.05. So we'll be doing these pairwise comparisons with all our different extremes, and basically looking out for candidates that keep appearing in cell lines with highly correlated phenotypes. So in conclusion, we took a holistic data analysis approach, considering the various aspects of the host-pathogen interaction, giving additional confidence in characterizing our extremes. We saw significant differences between the phenotypic spectrum of the assays. There is a correlation of cell types at the most and least permissive ends of the spectrum, and so between late and early events, as well as between HIV types. And we also found differentially expressed genes between the cell lines with extreme phenotypes that are statistically significant from one another. In terms of future work, we still need to finish conducting bioinformatic analysis on all of the RNA transcriptomes, integrate the genomic data with the infection data to identify additional potential regulators, and functionally test any candidate genes. So in conclusion, I'd like to thank you, my supervisors, my lab, my thesis committee, collaborators, funding bodies. And I also want to make special mention of one of my colleagues, Natalia Almeida dos Santos, who contributed massively to the data analysis aspect of this project. And I hope you got a chance to see her poster in the first poster session, which dealt with the iPSC-derived macrophages I mentioned earlier. Thank you.
Thank you, Neo. Great talk.
Thank you.
I'm sure there are a lot of questions.
Thanks for the nice talk. Did you check to which extent CD4 and CR4 expression just contribute to the different susceptibility?
So these lines do not express CD4 or CCR5 receptors. All of the vectors and virus we challenged with had to be VSVG pseudotyped.
Okay, but does this imply you are not measuring spreading infection here, but single around?
So essentially what we did was when we infected these cells, the supernatant that we took off them, we placed on TZM cells, which then measured the progeny infectivity, which we left for about 48 hours.
Yeah, very impressive, the amount of iPSCs you can carry.
Thank you, it took a while.
My question is, so when you create the iPSCs from blood or fibroblasts, you basically wipe the epigenetics of the cells, and epitranscriptomics as well, etc. So do you also have a baseline of how the cells started with? For example, if you have the PVMCs, could you do the same kind of things, or did you guys try within the 3 projects to just know if they relate to the original donor, like multi-infectivity or whatever you might be measuring?
So the genome is the genome, right? The genome doesn't necessarily change, but we would have to check with the actual collaborators who own the biobank to see whether or not they kept any of those samples on hand. What I can say is that we have transcriptomes from when those cells were first generated. So these would have been when they were reprogrammed, but not necessarily established iPSC lines. It takes a few passages before they become very consistent in the amount of spontaneous differentiation that they exhibit and stuff. So we could do some comparison, let's say, with the archive samples that we have versus the samples that we took from the time of infection. We could look into that. Thank you.
I was wondering, could you give us an idea what this culture conditions are like? And I mean, how it seems like, I don't know, an amazing feat to have 150 IPS lines. Are these colonies you're infecting, or do you disperse the cells, and how do you normalize these cultures?
Right. So they initially wanted 400. I did as many as I could. So it was a bit of a feat. It took me 18 months to screen all of these lines. Some of them didn't attach. I had to repeat certain lines and so on. These are adherent cell lines. They grow in colonies, which is the best way to grow them. Otherwise, they're not very happy. The way I describe these lines, have you ever played a little video game called Tamagotchi, where essentially it's a little It's a little device. It's a little electronic pet. If you don't feed it in like, a day, it dies. That's what these cell lines are like. So in terms of when we actually prepared them for the screen, we would break them up, dissociate them into single cells with ROCK inhibitor to aid their, attachment upon just before infection. Yeah, does that answer your question?
I don't know. I mean, there's so many questions about cell cycle and viability percentages. Do you monitor all these things?
So we monitor all of those things. To be honest, after we've got a countess that helps us, make this more high throughput, so that we can, we get numbers for all these cell lines in terms of what we plated, what the viability was. But to be honest, after a while, you get good enough, and the cells that you're working with well enough that you can basically assure 90% viability the entire time. In terms of how we normalized across them, like I said, we used a Z-score normalization to the week that we screened them on to mitigate the batch-to-batch variation that we might see. Initially, we did use another control instead of along with our 293Ts, we had an iPSC control that we ran with each run through of the screen with all the other lines that kept changing. However, this turned out to be just as variable as all the other lines, so it's not exactly the best control, hence why we decided to move towards Z-score normalization. Yes.
So nice work. Do you correct for ethnicity differences, or do you see any association?
So that would be one of the limitations of the project, I would say. We do have data to say whether it's the person that these cells come from is male, female, and general nondescript data, non-personal data about these lines. However, most of them come from the region around Cambridge. So if we were to approach this study again and be with knowing now what we do, I suppose if we had access to a biobank, efficient access, let's say, in terms of this, it was easy to get hold of these lines, which is why we opted for these. One of the reasons anyway. So using a more diverse pool of stem cells would be advantageous for sure.
Okay, then this was the last speaker. Thank you again, Neo. And
thank you, everyone.
Thank you, everybody, for the nice discussion, and we reconvene in 15